Hello, everyone. Thanks for joining. So today we've got Yang Hu with us from the University of Oxford, and he's going to be talking about interaction mechanisms between AI and medical research. So just some housekeeping. If you're here in person, please register on the tablet. The webinar will be recorded and made available online. Online attendees, please stay muted during the presentation, and there'll be some time for Q&As at the end. So just a bit about Dr. Yang Hu. He is a postdoctoral research scientist at the Big Data Institute at the University of Oxford, and he specializes in medical image processing, multi-omics analysis, and human-centered AI for our healthcare applications. Okay, should be okay.
Okay, thank you very much for a kind introduction, and thank you everybody to have me here. Today I would like to talk about, today the topic I would like to present will be slightly different with the initialized setting up, because recently from some preliminary work we find some very urgent concern I have to promote, I think, in the community, and is about what kind of trustworthy interpreting mechanism we should build if we would like to connect the human and AI model in our future applications. So I think I will skip this page because Sarah has already give us a very nice introduction about me. And in case we have some audience from the background of not computer science, I would like to take a few minutes. I would like to take a few slides to introduce some very basic knowledge about AI and computer vision model. So here's the example about how the AI model deal with the image in the deep learning framework. And usually in the eyes of the AI, each image will be represented as the format of the matrix, and you can see as in the border, different value will help us choose the characters on the image. And the neural network will feed forward the signal from that input image and finally output it and compare with the annotated label. And if that's incorrect, it will feed back. It will feed back the back propagation loops of that. So after that, we will collect usually in the last layer of the phonic in the neural network. We will collect the feature vector, so that's kind of the compressed signal for the input images. A more popular computer vision model is a convolutional network, neural network. But now we have something more advanced model like vision transformer and member, or some even KAN. But the convolutional neural network is still quite popular for using, and you can see from the different deep layer, we can extract the feature more and more abstract to describe the input images. And but today I would like to show some case study on computational pathology. So the image type we are dealing with is quite different with the normal images. That's whole slice imaging. So the whole slice image, the collection pipeline of the whole slice image is like that. Usually we just collected the tissue from human in the process of the biopsy and surgery sometimes, and we just scanning it on the slide and make it digitalized, so that it's quite convenient for us to viewing and manage and sharing with the other people and do some analyzing on the computer side. the problem of the modeling process of the whole slice image is that this is not the typical type of the image. It's quite large. It is extremely high resolution, and which can be split into many small patches with the normal size of images. So that means we cannot dealing with that with the existing memory available. We can on the devices we can get. So the usually practicing is that we use a specific machine learning algorithm called multi-instance learning to deal with the whole slice image. Usually we will divide each whole slice image into a lot of small patches and to process, and we will sharing one label of that to help us training a model to do some prediction. That's based on the setting of weakly supervised learning. So that means maybe only a very small area on the whole tissue is quite useful for that specific task, and the other area is irrelevant. We think is relatively noise or background area. So in the first use case, in the first study I would like to present is about, we find that the fine-grained feature on the small patch level will help us driving the evolution of tissue level morphological understanding. So what is that is the task we are going to accomplish in this study is that the molecular analyzing based on the histomorphological features. So what is that? What is the molecular subtype? So as we know, tumor is a very dangerous disease for most of the patients, right? And the tumor subtypes based on the molecular features actually is quite important for to guide the personalized treatment for different group of patients. And but the problem is the molecular features rely on the sequence analyzing equipment, and it's very expensive. So for example, if we would like to help NHS save money, we need to find a cheaper replacement from the perspective of the digital pathology image analyzing. So we did some very remarkable preliminary work in about 4 or 5 years ago, and we are the first group to test only use the digital pathology imaging to try to predict the subtype, molecular subtype in the colorectal cancer. So we just designed a neural network framework, deep learning framework to training the model and try to encourage it to predict the four molecular subtype in colorectal cancer. And we find that across after validation across cohorts from different hospitals, we find that the performance of that currency performance, it looks quite promising, and we are quite excited about that. So we decided to expand some similar pipeline in some other type of cancer. For example, we collaborate with GR Hospital in Oxford and do some prediction for the EMT, high or low for the ovarian cancer, and which indicated very serious, very poor survival rate. So the challenge we are facing in this study is that because usually when we would like to train a deep learning model, we are rely on quite big cohorts But as we also know that the collection process of the clinical data is quite difficult, and so in this case, actually, we only have 70 of them. Can I minimize that? we only have 70 whole slice images on ovarian cancer, and we still need to split it into training and testing sets, right? So that means we only have not more than 50 of them for training. So I think based on given that situation, the typical multi-instance learning pipeline will not such smoothly work. And let's just have a quickly review about the multi-instance learning pipeline. After we separate the tissue into many small patches, we will use a pre-trained feature encoder to extract the feature of that each patches, and then we just feed all these compressed features into a field layer fully connected neural network, and we call that aggregator, and to give us the final prediction result. And also we will set up some attention mechanism to help us have the attention map show so that we can indicate which area is more important for that prediction. But the problem is, so after we extract the feature from for each patches, actually we don't have the opportunity to further optimize them. That means even we find which maybe that area is more important for this prediction, we still don't have the opportunity to retraining them and have a somewhat better features for this prediction. So based on given that situation, we just proposed a method called self-interactive multi-instance learning. So it's quite easy. It's quite simple framework, and which consists in 3 steps. The first is to select the high attention types from typical attention network that aggregate and fine tune, and so that we assume these high attention patches are kind of representative features, representative types in the whole slide image. And then we just fine tune the tie level features with these high attention types, and we just assign the same label with the aggregator. And then we refresh the tie level features for the next round of attention neural network training. So this work was published in the earlier of this year in Medical Image Analyze. So we get a very impressive improvement on the accuracy, but I think it's not a most important part of it of our result. So actually, we find that we find some summarization about, as I just mentioned, the fine-grained features optimization. Along with that optimization, the overview understanding of the whole tissue will be also optimized. As you can see, for those informative areas, they are gathering more and more attention. For those background areas, actually, the attention on that has been described effectively. So, and also in the feature space of the tie level images, the distinguishability of that are also be optimized so that we can setting up the aggregator in a much easier scenario to conduct the prediction. So finally, we can have a very good region of interesting detection ability only based on not more than 50 training samples, while the other method usually request more than 1,000 slides. So here for this study, we have some takeaways. I think the first, of course, we just reduce the data requirement from 1,000 to not more than 100. Another very important takeaway is that because we need to provide these interactable results to the clinicians, to the human, I mean, and ask them to check, okay, if you think the understanding of the AI model, if you think the evidence provided by the AI model makes sense to you or not. Actually, we will show them the 5 grand features. If the 5 grand features are optimized, it will impact on the global tissue level interpretable performance, so that we have a more efficient visualization strategy can provide some more expandable result for our clinician collaborators. But recently we have some new results shows maybe they still have some potential dangers between in that. So before we construct an interaction pipeline between AI and human, I think the first thing we need to build up a very stable, reliable, trustworthy system from the evidence providing aspect of the AI, right? So, but now the people in the community actually pay much attention on accuracy. So I just raised this question that the high accuracy really means better. So in this study, the very, very new study, and we haven't published yet. So we actually, after we submit the previous work in 1 or 2 years ago, and actually that's a very game-changing project for computational pathology. So in last 1 or 2 years, more than 20 pathology foundation models have been introduced. So what is the foundation model? So these foundation models are based on very big cohort invested by the big company or top university from America. And they just pre-training the model on a lot pathological images and encourage the model, just assume the model actually can see almost all kind of the images. So they assume this foundation model actually have a very powerful understanding about all kind of the pathology images. But up to my investigation, we find that all these foundation models actually rely on very basic data source. So although they collect a very big cohort, but most of them are just collected from 1 or 2 hospitals. And also they exhibit a very imbalanced cancer type distribution. That means although the cohort is quite big, and they claim the cohort covered about 20 types of cancer, but most of them are coming from few types of cancers. So, and as I just said, they focus too much on the accuracy, so that sometimes they will ignore the stability and the reliability for the interpretable result. So in this study, we just trying to monitoring the attention shifting behavior for different foundation models, so that from this perspective, we are trying to understand how these foundation model will understanding the downstream tasks. We just tested 2 markedly proven pathological foundation models on 5 downstream tasks, and then we just set up a comparative experiment. So first, we checked the accuracy of these foundation models. We found that these 2 foundation models actually perform the accuracy quite almost the same, quite similar on all the different downstream tasks. And then we just track the attention moving behavior to simulate the thinking trajectories of these foundation models during the downstream task transference process. For example, we just monitoring if the model will moving the attention from some specific area to some targeting areas. And in addition to that, we also identify the region with sharp local attention change to indicate a significant shifting in their neighborhood. And when we reviewing the patches with significant attention move in and move out, we just observed extremely tiny overlap in their distribution across different foundation models. And we also check the initialized value of those attention of the attention for those patches. We find that there is no reverse situation for the attention for them. They all the initialized value of the attention for these patches all come from about 0.5. And then we just do some task specific analyzing. And the first task is the ovarian cancer treatment response prediction. And by clustering the high attention movie in patches on these two different foundation model features, we can actually identify the distinct morphological phenotypes represented by each clusters, right? And then some of them shows very obvious overlay, but while the much more others attention moving regions actually display the entirely different phenotype. So we expect the consistent it will show some consistency in the morphological understanding across the different foundation model. But actually the realistic situation is that it's quite hard to reliably capture it. And the different local attention shift pattern also uncover the distinct morphological understanding when we applying these two type of foundation model. Another task is molecular subtyping of colorectal cancer, as that cohort we have done the previous work. So we just locally deploy the DeepSeek large language model to help us generate some description about those attention moving areas from different foundation models. We found that for both attention moving or move out areas, the description about phenotype of those foundation models shows for some of them, I even cannot correctly, I'm not professional in medical science, I cannot pronounce it currently, but obviously you can see it's quite different. It shows the description about those phenotypes is obviously different, right? Another task specific analyzing is on marsh grading of the qualities, and the description of the phenotype from different foundation models also shows very significant heterogeneity. A more interesting result is that when we combine all these multiple data sets and the clustering the attention move in or move out area together, and when we test one side, for example, when we test the attention move in clusters on this side, on the to the other side, we find that the cluster of attention moving patches shows much more significant similarity with the attention move out cluster in the other side. So it's not only significant inconsistent in the attention shifting behavior. Actually, that's sometimes even opposite tree in the attention moving behavior. So this is the one more ongoing work. We are also checking the cell level attention moving or move out situation on the tissue. So, and this is some map after we check each cell, how many of them just have the attention moving, how many of them just have the attention move out. So I would like to have the conclusion and stop point with some open questions. So is the trustworthy of the AI in medicine, is that still a vision, or we are almost approaching it? So I would like to express my opinion with all these block points, all these valid points. The first is the foundation model looks like it's superpower, actually, but I think actually that brings a very urgent need for the reliable interpretability. Another is that actually not only for computation pathology, some other foundation model also have the same experience. Many foundation model actually drop even very basic attention-based explanation in favor of recurrency. So I don't think a very good idea because it's a dangerous trade-off for the clinical using. And even we have the attention-based methods, as we know, the attention-based method alone cannot fully uncover the model thought. So maybe we need to explore some more advanced interpretability approach to and beyond attention. So another very hot topic recently is about multi-agent system. So just imagine that in some near or long future, when we really want to introduce in the multi-agent system in our clinical workflow, it may raise some new risk. So the agent will developing their own secret language to bypass the human's oversight. So if that, how can we just construct a robust safeguard to avoid it? Well, it's still quick. So this is my presentation today, as I think I left it much time for discuss.
Yeah, that's fine.
That's great.
Yeah. Hi.
Sorry. For your attention heads, what is the actual algorithmic methods you actually use in the algorithms? The various, other different ways you can do attention heads.
So for the attention mechanism, we just set up a layer to encourage it to assign the different weights for the different region.
Yeah.
And it's also embedded into the whole continue feed-forward pipeline. So when we optimize the whole network, that will assume to be find the optimized attention distribution on different spatial regions.
Yeah. What are you optimizing though to find the weights in your attention head? Is it just some sort of cross-entry?
So finally, we will have because this is the classification task.
Yeah.
So finally, the loose function will be cross entropy. Yeah. Hi.
Yeah, I was a bit unsure about like multi-instance learning. So I understand the aspect that you have a really large image or the slide with billions of pixels and you make them small patches. And then, but do you parcel the patches in the model and then form a loss function, or is it like contrastive learning where you have a different patches and you're trying to maximize similarity? Like, could you explain the multi-instance function a bit?
Yeah, this is not contrastive learning because it's a relatively end-to-end pipeline, and the only thing is that after we divided the whole slide image into many small patches, we just assign. So for example, we just assign the label for the whole slide to actually we just assume all these patches share one labels of that. Yeah.
Like what sort of loss do you use? The classification loss?
Yeah, classification. Some normal classification is the only thing is that that's why we need to apply the attention mechanism because for the whole tissue, not all of them are informative for the specific task. Actually, most of them are background area, so it's not useful for learning. So we must assign the different attention for the in the spatial areas, so that highlight some of them. Try to encourage the model to highlight some of them. Yeah. Hi.
Following this exact point, Do you assign then binary labels, or do you reduce the strength of the labels? Like, for example, stop using soft labels, given that you know that the majority may not contain cancer, for example, then you may reduce the output. Instead of one saying cancer, you may have the prior probability given the number of slides that have cancer.
Yeah. So that depends on different tasks. For example, for some specific already detected subtypes, the clinicians will suggest us to set up a very hard label, like yes or no.
Yeah.
So for example, for KRAS, if that's KRAS or wild type, so just a relative. I think that's a binary classification, but it depends on different tasks. If we would like to, using some generative pipeline to generate the expression of genes, that will ask us to set up a more complicated output layer, like some continuous sequence layer about gene expressions. Yeah, it's totally depend on different tasks.
Yeah, but what I meant is that in the multi-instance learning, you have a huge image.
Yeah.
You divide it into patches.
Yeah.
And each of these patches you assign the label, for example, cancer, because there is cancer in the full image. Then what I mean is that for weak learning, sometimes you have soft labels. Instead of saying that there is actual cancer on that patch, you actually have a lower probability. But
as I said, it's also depend on different tasks. For example, if we would like to predict the survival rate, we can encourage the model to decide it. Well, we cannot say that. So although we can setting up the output like a continued value about how much percentage probability this guy will die, but we cannot assign that on each specific patches, because we cannot say, oh, from the perspective of these patches, this guy is very likely to die. For another patches, this guy is very hopefully to survive. It's not that case. Because that's all the tissue is coming from one person, right? Yeah. Hi.
I didn't fully understand the attention shifting aspect. What is shifting? Like the attention as you're training using the foundation model, or the attention as you go along the different patches in the image?
So in that case, we're just setting up 2 time points. The one is before we fine-tune the foundation model's features on the specific downstream task. Another one is after we fine-tuned it on the specific downstream task, and we can observe the distribution of the attention map is quite different. So maybe they just moving some attention initially on some stronger areas to the tumor areas. That means, oh, after learning, the model actually is try to understand more about the tumor, right? So that's a very intuitive ideas about monitoring those AI models thinking pattern when they are facing some specific tasks. Yeah. What?
I had a question in terms of your labeling. Sorry. So in terms of like the clinician like label, was that for the whole image, or was that like specific areas of the image?
We very hope clinicians can help us label some specific areas and tell us, okay, this area is like that, this area is another label. But unfortunately, all the pathologists, all the clinicians don't have too much time. They still need to respond to the duty of the clinical side. So they can only provide us, okay, this case is KRAS, for example. This case is Y type. So we have to, they just hope us can develop some more advanced technology like that to indicate that area is very useful for determining if that's KRAS or Y type. Yeah.
You must be prone to outliers in there when clinicians actually simply got it wrong, put a bad data point in there. Do you have strategies to actually try and detect outliers or learning them?
Wow, it's a very interesting question. Can you explain more?
Yeah. I mean, one way of doing it is you have very reliable data points. So you learn off these, and you then sequentially learn data points.
Yeah.
Each time you predict ahead what you think it is with a confidence measure. And if it's simply the opposite, a high confidence label, it could be an outlier, and you may simply not want to learn it. So it just goes with the reliable data points. If you do that, it can actually bounce up performance.
I think we haven't do something like that yet. I think it's quite a good idea to try it, maybe. Yeah. Thank you.
I just wanted to add on with your point that all players may have actually high.
But you don't rightly know, because it could be rather unusual but correct. Or an outlier.
Yeah.
But I've done experiments where you sort of with support for correct machines, where they're sort of like completely on the wrong side, and there's a point you go through a dip of free test error, and the other side it's not profitable to learn these, even though they could be unusual correct. In fact, the probability that they're actually outliers is too destructive. So you typically go to a little dip, and then you start going up again, dropping your accuracy, test accuracy, because you're deleterious now.
Oh, interesting. Okay.
I'll just go one line.
No, I
There's
don't. We can keep an eye on this chat for you, so don't
Yeah,
don't feel like you have to have this open. You
Sure.
can get your slides back up.
So on one of the slides, there was a brief comment about using domain adversarial training for classification, I think.
Which one?
It mentioned like classify performance with domain adversarial training, I think. It was a quick slide that went over.
This one?
It was a table with results. I think this one.
Oh, this one?
Yeah. Did you explain this one a bit more? And what was the purpose of domain adversarial training?
So this means we just tested on three cohorts, and just for each specific subtype, we just also calculate the currency about that.
Okay, the domain adversarial, like I don't remember too much about the domain adversarial training, but does that like make it harder for?
Yeah, adversary training is that because we would like to encourage model have a stronger generalizability, and for example, I think here, okay, for example, generalizability, because all the staining and the background of the scanning, yet some noises on the artifact, on the tissue, on the slide, they all have different representation. So when we training one model on one cohort, we want it as generalized as possible and can be directly applied on the other cohort. So we just setting up advanced retraining mechanism that encourages the model also predict each samples coming from which cohort, and we just set up opposite loss function feedback to encourage the model to ignore that part.
Okay, so it would be like sort of using data augmentation in the data in some way.
It's not data augmentation. It's just for example, we just it's a multiple task system, and one is our main task about subtyping classification. Another one is ask the model predict which cohort of this case belonging to.
Okay.
For that task, we just encourage the model has as bad performance as possible.
So to have performance on knowing which task.
So that we just encourage the model try to ignore, not be biased too much on different cohort.
Okay, and how will that work? Did you have like random labels for that task?
No, we just setting up a negative loose back propagation. So for example, for the main task, we always will feedback our loose in the, let's just assume it's in the positive direction. For that side task, for that advanced task, we just set up the loose function as a relatively negative direction to feedback, so that we can encourage the model try to ignore that part of the image, that part of the information. Yeah. It's quite a difficult, it's quite a very detailed technical. Yeah. Hi.
When you were comparing different foundational models, I believe,
Yeah,
you showed that they have similar performance, but they had different
an interpretable performance. Yeah.
areas of interest or
Yeah.
And maybe I missed that bit, but was there any conclusion of why they were focusing on different parts, and were any clinicians actually trying to understand the different reasons?
No. We are planning to do that in future, but for now we haven't done too much investigation about the in-depth reason behind it. So, but actually, I think it's quite exciting result, because we can, at least we can raise some concern in such a situations about a lot of people are presuming bigger and bigger foundation model and presuming higher hand accuracy. Maybe they should have some new perspective to understand the whole picture of the problem. Right.
And I think it also shows the potential of ensemble models, because usually you want ensemble models to provide you different views.
Sure.
Even if they agree on the prediction.
Sure. Yeah. That's one another work we are doing now. And we actually, I think if that's possible, I'm really happy to present again in the second half of this year. And I think at that moment we will release some more research work about that part of research, that part of study. Yeah.
When you're saying that you only have 49 samples, 59 samples, or 49 samples, 50 samples, and that was low amount of data, how did you make it data efficient? I didn't fully understand that.
Yeah, so here we just have so given we just have not more than 50 samples, whole size, which looks like is the cohort is quite small. Yeah, indeed it's quite small, but don't forget we have for each slide it actually is quite big. If we separate them into many small patches, actually we can still collect a quite big data set in the tile level, in the patch level. So the idea basically is that how to very efficiently leveraging the feature in the tile level. So if we can using the feedback information from the slide level to optimize the feature on the tile level, we can make the tile level features more efficiently contribute on the slide level prediction, right? So that's a basically idea.
And how did you optimize it for the tile level? And also, isn't there issues of independence, because the samples all correlated if they come from the same person, right? Like, is there would it be redundant information in because all the patches from one slide are related to one?
Yeah. So as I just said, that in the typical pipeline, we don't have any opportunity to further optimize the tile level features. So the tile level feature actually is extracted by some frozen feature extractor, and which was pre-trained on somewhere, maybe foundation models, and in our framework, we just selected those high attention patches as the representative patches for each slide, and using them, we assign the same label for them to fine tune the tile level feature encoders, so that we have opportunity to further optimize that on our specific task. Yeah. Yeah.
The original images are huge, and then you decided to divide them into batches.
Yeah.
Because that's computationally feasible to train a convolutional neural network on those batches with lower resolution. No?
Mm-hmm.
But there are other approaches, like having a hierarchical representation, no? Like reducing the number of pixels, aggregating the number of pixels, no? At different scales.
Yeah.
Did you try that as well?
That kind of hierarchical framework was proposed by Harvard University, I think, and they proposed some similar framework like that. But the drawback of that framework is it will cost too much computational resource because they have a very big computing center and they have a lot of GPU.
Yeah.
But actually this framework actually can apply it on one card scenario. Only need one GPU card.
Okay.
Yeah. So so that means we don't have to storage too much hierarchical different scale of the features in the memory.
Yeah.
Right. So
yeah, but I was wondering, given that you are using an attention mechanism, then maybe having the whole picture with low resolution may already select which areas to look at. Then once you select those areas, then you can zoom in and then do again the attention mechanism and then until you get the predictions.
Indeed, yeah, that's doable, but usually we assume sometimes the resolution losing will cause some uncertainty maybe.
Yeah.
That will systematically cause the following mistake. Yeah.
Potentially. But I can imagine the original image having lots of background.
Yeah, have a lot of
just white.
Yeah, of course. Yeah, of course. We
which
will
is not interesting at all.
Yeah, we will. Yeah, in the pre-processing step, we will just discard the part.
Oh, I see. Okay.
Yeah. And okay, I think it's quite nice. Yeah, we still have 10 minutes.
I mean, we can finish. And keep the.
Yeah, thank you very much. Okay, thank you very much. Thank you for having me here, and it's quite honored. So if I have some update about my new research, I'm very happy to come here again and represent. Yeah.
Okay, thank you.